Okay, so in the previous lecture, we were talking about hydrogen bonds, the non-bonded interactions, right? And we were talking about wherever hydrogen bonds are present, they are so strong that they are responsible for the, tertiary structures of proteins. They are responsible for binding of many ligands with the receptors. And the strongest of hydrogen bonds are often formed between the groups with greatest electrostatic character. For example, carboxylates. Okay, so carboxylates and a COO minus is carboxylate ion, right? So wherever we have COO minus as a part of the structure, they can very easily form a hydrogen bond. Whereas the other groups like amides, ketones, and unionized carboxylic acid groups, they like a COH group, they may not form that good hydrogen bonds. Then, on the other hand, we have ammonium ions, which are better donors than unsubstituted ammonium ions or trigonal donors. So, instead of NH2, if we have NH4 plus, okay, then that will tend to be better in terms of hydrogen donor capacity and forming a hydrogen bond with CO minus as compared to many other groups. Okay. So talking of the steric interactions, then we have those short range repulsive forces. So we just talked about van der Waals forces of attraction, or the which are also referred to as the London forces or dispersion forces. Now, when a, a ligand comes very close to its receptor, then what will happen is there will be repulsive forces that will start to creep in, and that will make the ligand to move away. Okay, and hence there is always a optimum distance that is maintained between the ligand and its receptor. Okay, and this equilibrium distance can be determined from X-ray crystallography, from the crystal data. Okay, so this is nothing but equivalent to the sum of the van der Waals radii of the two interacting atoms from the ligand and from the receptor. So if we have a atom from the ligand and another atom from the receptor, and then they are coming together, so whatever is their van der Waals radii, उतने डिस्टेंस तक ही दे कैन कम क्लोज. उससे ज्यादा अगर क्लोज आ गए तो they will be repelled, and that is what is short range repulsive forces. Okay, then for non-polar molecules, this balance between the attractive dispersion forces and the short range repulsive forces is generally defined in terms of Buckingham 6 exponential equation, which is written over here. And then there is another equation which is called a Lennard-Jones 612 potential, which is written over here. Okay, so it is called as Lennard-Jones because it was discovered by those, it was put forth by those physicists. And 612 isliye because we have those terms 12 and 6 in the equation. Okay, so these are two important equations, especially this one, Lennard-Jones 6-line potential, 612 potential. This is a very important equation in calculation of the short range repulsive forces. Okay, so this is something that we often use in many of the computational softwares. Example, QSAR. Okay, for three-dimensional QSAR, when we have to talk about the steric component, we'll use Lennard-Jones potential. Okay, and the extreme repulsive forces that develop as atoms approach closer than the sum of their van der Waals radii are perhaps the single most decisive influence on whether a molecule will bind to its receptor, since very small steric clashes between the molecules will abolish affinity. Okay, so जब ligand and receptor एक दूसरे के पास आते हैं, तो whether they will actually bind or not, that will be actually determined by these short range repulsive forces. Okay. Then, so we have just talked about the enthalpy contribution, the different types of interactions that happen between the ligand and its receptor. Now, let us talk about the entropic contributions. Entropic contributions can be in terms of translational and rotational entropy. So, the formation of any ligand receptor complex is accompanied by the replacement of three rotational and three translational degrees of freedom of the ligand by six vibrational degrees of freedom in the complex. इसको हम कहते हैं residual motion. Okay, to understand this thing, when a ligand and a receptor binds, three rotational and three translational degrees of freedom of the ligand are replaced by six vibrational degrees of freedom in the complex. Okay, and the extent of this change is primarily dependent on the relative tightness of the resulting complex. So better the more tight the complex would be, better would be the binding, right? So for a typical ligand protein complex, estimated change in free energy resulting from loss of entropy on binding at 310 degree Kelvin ranges from 9 kilojoules per mole for a loose interaction to 45 kilojoules per mole for a tight interaction. Okay, so if we are talking of a ligand which is very loosely binding to its receptor, the interaction, the loss of entropy energy on binding may be as low as 9 kilojoules per mole. Whereas for a ligand which is binding very tightly with its receptor, the loss in entropy may be as high as 45 kilojoules per mole. In addition to this loss of rotational and translational freedom, there is further entropy loss due to conformational restriction that accompanies the binding of flexible ligands. Now we know that ligands are flexible. We know the receptors are also flexible. So जब ligand and receptor bind करते हैं, the flexibility of the ligand is lost. Okay, the flexibility of the ligand is lost. And in case of rigid analogs, for which there is no loss of conformational entropy on binding, this factor provides the free energy advantage relative to the more flexible ligands. And this is the basis of one of the central tenets of medicinal chemistry that is called as conformational restriction. Okay, so medicinal chemistry may in your undergraduate years you must have studied about conformational analysis, conformational restriction, which conformers are binding, etc., etc., right? And that is one of the basic concepts of medicinal chemistry. So the ligands need to be flexible so that they can flexibly bind to the receptor. But on binding, they will lose out on their degrees of freedom. Okay. So conformational flexibility is lost. It is referred to as conformational restriction. In some cases, the bound conformation of a flexible molecule may also be its lowest energy conformer. Okay, in which the penalty for which the resulting conformational constant will be restricted to the loss of internal rotational entropy only. So, if it is a low energy conformer, then it is better because वहाँ पे penalty in the calculation would be only due to internal rotational entropy loss. And in other cases, the optimal interaction between ligand and receptor will require higher energy conformation, and this energy penalty will reduce the net binding energy that is observed. So if at all higher energy conformers are binding, then the binding energy decreases. Okay, if higher energy conformers are binding, then binding energy decreases. In other words, binding affinity decreases. On the other hand, if low energy conformers are binding, so yesterday we had talked of a potential energy curve, right? So we talked about the global energy minimum and then few other energy minima. So if this particular conformation is binding, that is best for binding. If this is binding, then it is okay as compared to this or any other conformers, right? So if high energy conformer is binding, then the binding affinity decreases. If low energy conformation, especially global energy conformation binds, then the binding is better. Okay, and that is the reason why we need to do conformational analysis of all the conformers that are possible on a potential energy curve for a given molecule. The lowest energy conformer is always involved in binding. Okay. Now, hydrophobic effect. Now, when a non-polar molecule is placed in water, strong water-water interactions are formed around the solvent molecule to compensate for the weaker interaction between the solute and the water. Now, imagine we are placing a molecule, a ligand, and it is in water. Okay. So, what will happen? Water, because it is not able to solvate this molecule, what will water molecules do? They will arrange themselves around the ligand in a particular manner. Okay, and this arrangement is nothing but entropy change, right? जब वाटर मॉलिक्यूल्स फ्रीली मूव कर रहे थे उसके बीच में हमने एक ligand डाल दिया and what is happening with this ligand or this drug now, it is now very nicely I mean it is causing the water molecules around to very nicely arrange themselves around it. Now that is nothing but loss of entropy. Entropy, in other words, is the orderliness of the system, right? Or rather, the disorderliness of the system is what we talk about when we talk of the term entropy. So, जो disordered system था, disordered water molecules थे, they are now ordered. That means there is a loss in entropy. Okay, so this results in an increasingly ordered arrangement of water molecules around the solute and thus a negative entropy of dissolution. Thus, the decrease in entropy is roughly proportional to the non-polar surface area of the molecule. Okay, so that means if the surface area of the molecule is non-polar, then there will be a it will be directly proportional to the decrease in entropy when the molecule is placed in water. Okay, and then the association of two such non-polar molecules in water reduces the total non-polar surface area exposed to the solvent, thus reducing the amount of structured water and therefore providing a favorable entropy for association. Now imagine water जो disordered था उसके बीच में हमने solute डाल दिया, I mean अपना hydrophobic ligand डाल दिया. So what is happening? Water is arranging itself around this hydrophobic ligand. Now suppose this ligand approaches the receptor. So what will happen? Receptor is also kind of hydrophobic. Ligand is also kind of hydrophobic. So instead of arranging around the ligand, now this water will start to arrange around the receptor and the ligand complex. So in other words, what is happening? In other words, the entropy of the system that is going to be decreased, and orderliness that is going to be induced will be decreased because there are two molecules, or rather the receptor and a ligand, which have come together now. So that is going to increase the association. That is going to increase the favorable entropy of association between the ligand and the receptor. So this is called as hydrophobic effect. Okay. Although it can be determined theoretically from statistical mechanics calculations or free energy simulation, the hydrophobic effect is most commonly estimated empirically. Empirically यानी tables use करेंगे जिसके अंदर we'll have different functional groups and their hydrophobic effects. And then what we'll do is we'll just look at what all functional groups are present in the given ligand and its receptor, and then we'll just add those values up. Okay, so when we add those values, what we'll get is the hydrophobic effect. Okay, so that is what is meant by the hydrophobic effect is commonly estimated empirically, यानी कि calculations करते हैं hydrophobic effect. Okay, one of the methods used in the measurement of solubility of simple hydrocarbons in water is solubility of simple hydrocarbons in water, that is solvent transfer method. And estimates of free energy required to transfer hydrocarbons from water into hydrophobic environment range from 0.1 to 0.14 kilojoules per mole for every square Armstrong of solvent accessible hydrocarbon surface removed from the exposure to water by the binding process. Now let me just remind you all once again that we don't need to remember all these values and all. Okay, our idea is to understand what is the basis of a ligand interacting with its receptor. How can we design newer ligand which can be very nicely and very tightly binding to the receptor? Okay, so all these values are just for your understanding. Okay, please don't get stressed looking at those values. Now, solvent reorganization is another thing that is happening. So similar to hydrophobic effect. Now, when we place a ligand and a receptor, तो receptor के आजू बाजू में जो water molecules था, they will be displaced by the ligand. Okay, so that is nothing but the solvent reorganization. Okay, so in principle, the entropic component of the hydrophobic effect described in previous section might be applied to broad range of hydropathic types, not just non-polar molecules and surfaces. Okay, so in 1997, a scientist called as Claude, he made a theoretical derivation that electrostatic interactions, not only ionic, in water would have large favorable entropic components. So he said that अगर ionic interactions होंगी या electrostatic interactions होंगी, तो they will have large favorable entropic components, and this may be rationalized as similar to entropic components of the hydrophobic effect. So अगर hydrophobic components है ligand में यानी hydrophobic functional groups है, they are going to cause hydrophobic effect. Whereas if the ligand has hydrophilic components, yeah, ionic components, then that is going to cause solvent reorganization. Okay, so the solvent shell of the polar groups would be more ordered than the bulk solvent, and an interaction between two such polar groups would eliminate some of the water back to the bulk of a solvent with favorable entropy gain. So this is nothing but what we have just talked about. Okay, so when a hydrophilic molecule or a ligand with hydrophilic functional group is approaching a receptor, then the solvent, the water that is present near the receptor, will be displaced by these hydrophilic groups, and that water will be thrown away from the receptor. So that is what is mentioned over here in technical language. Now, Klotz, this researcher Klotz, he used the dissociation constant of acetic acid in water as an example, and he went on to calculate many different functional groups, many different hydrophilic functional groups, and their effects on binding of ligands to the receptor. Okay, so negative entropies of transfer of ions and molecules from the gas phase to water also support this particular effect, that is solvent reorganization effect. So this is seen not just in solution but also in gas phase. Okay, but also in gas phase. Correct. Then let us talk about the next concept, that is total energy of intermolecular interaction. So we said that there are methods for calculating the intermolecular interaction, and for the first method is free energy perturbation. Okay, so free energy perturbation, we had talked about it in the previous lecture. Free energy perturbation techniques are used to determine the difference in the free energy of binding between two ligands and a common receptor, or between the same ligand and solvent, by utilizing the concept of thermodynamic cycle. Okay, so simply put together, what I can say is, sorry, if we have a single ligand, or sorry, a single receptor, and we have different ligands. So sometime back in a previous lecture, I talked about naphthalene. Okay, so structurally related structure compounds which have only small substitutions which have been here and there. So if these are made to interact with the receptor, okay, if these substituents interact with the receptor, then what would be the change in the free energy of binding? Okay, so differently substituted common structure and its interaction with the same receptor. Okay, so that is called as free energy perturbation technique. Okay, so this method has proved astoundingly accurate in predicting relative binding energies of ligand series, but still suffer from problems of computational expense. So even though these are very accurate calculations for a given series, yani ki related structures of ligands, but still they are computationally expensive. And its use is limited due to narrow structural changes between the two ligands. I'm saying that this naphthalene in the first structure had chloride over here, and in the second structure it had a methyl group over here. Okay, or maybe some other alkyl group or aromatic group over here. So the change in the structure is subtle. Okay, many times subtle, and that limits the usefulness of free energy perturbation technique. Okay, it does not require any partitioning of individual energetic components for the free energy change. So that is what is free energy perturbation. Another method of calculating the total energy of intermolecular interactions is the partitioning methods. Okay, so partitioning methods may what is done, different structurally different molecules, they have been studied, and then their partitioning behavior has also been studied, and then that has been correlated to how they bind to the receptors. Okay, how they will affect the binding of the ligand to the receptor. Okay, so the total free energy delta G resulting from ligand receptor interaction is the sum of the free energy changes associated with all the electrostatic, inductive, non-polar, and hydrophobic interactions, and less any conformational energy, rotational energy, translational energy, conformational entropy cost. So whatever are the free energy changes that are induced due to electrostatic, inductive, non-polar, hydrophobic interactions. उसमें से if we reduce the or if we deduct the conformational energy and rotational, translational and conformational entropy related penalties, so then we get the free energy change, right? So some of these terms can be calculated relatively simply using the equations in the preceding slides. And other such entropy terms are more complex and are frequently handled empirically, again, using calculations, using tables wherein the partitioning values, the values for the different functional groups are mentioned. So depending on what functional group is present in our ligand, we can calculate as to what would be the kind of interaction that the ligand will have with its receptor. Now, in order, it is important to avoid double counting these terms, and the total free energy change can be calculated in a variety of ways. So if we have a functional group, okay, so it can have rotational, translational, conformational, entropy cost associated with it. And also it can have electrostatic or inductive or non-polar or hydrophobic type of interaction. So it is very important that we should not double count these terms. Okay. And ultimately, a complete theoretical description of all the energy contributions can be described, and that would require quantum mechanical treatment of the entire system, including solving the time dependent Schrodinger's equation for ligand. Now these are all too complex technical terms. No need to be stressed about these terms. Okay, just try to understand the gist of it. Okay, so we need to solve the time dependent Schrodinger's equations for ligand receptor and surrounding solvent, and then the resulting free energy of the interaction would be directly comparable and hopefully in close agreement with the observed dissociation constant for the ligand receptor interaction. So what we are talking about is calculation of KD using partitioning methods. Now it is doubtful, however, that there is real benefit for the this is of real benefit in the 3D QSAR or drug design studies for the calculation of intermolecular interactions due to high levels of complexity. Now we are talking of all different types of interactions, and then we are subtracting all those penalties for those different entropic losses. What happens is the equations become very, very complex. Okay. So the real question then happens to be what is the total electrostatic or van der Waals or enthalpic or entropic or hydrogen bonding or hydrophobic contributions to the intermolecular interaction, right? So rather we would want to know the answers like what is the increase in binding which I can expect if this functional group is added to my ligand. Okay, so instead of talking of all of these interactions and making, complex derivations, complex calculations, instead of that, can we just look at the receptor structure and say, if I substitute this particular group by some other group, what would be its effect on the total binding of my ligand with the receptor? Okay, my newly designed ligand with the receptor. So that is something that we need to figure out. Okay, and that is done using the partitioning method. Okay. And the last part of it, estimating the individual group components in ligand receptor interactions and their cooperativity. Now, this is something again related to the total free energy of interaction between the ligand and its receptor. Okay, so if we have to measure the strength of association between the two molecules, we need to although we do not know anything about the overall quality of their binding. Okay, then we need to observe the increase in interaction energy resulting from the addition of new functional groups that might be consistent with what we may have anticipated. So a good experienced medicinal chemist will have this tendency of anticipation. Okay, so two slides back we talked about the structure of naphthalene. What happens if we have a chloride over here? What happens if I substitute with that chloride with a methyl? Okay, now we know chloride is a halogen. Chlorine is a halogen. Okay. On the other hand, methyl group is a pure alkyl group. It will have a plus I effect. Fluorine will have a electronegative effect, right? So all these factors will come into the picture. So we just need to understand, look at those functional groups, and we need to try to grasp. We try to you know, kind of gain experience looking at the structures, looking at those different functional groups which are substituted, as to what will they have, what effect will they have on the binding of the ligand with the receptor. Okay, so that is estimating the intrinsic binding strength of the individual functional groups on the free energy changes. Okay, so instead of going through the entire slide, I've just given you the gist of it. Okay, so what happens when we change the substitutions? That is the functional group, one functional group by another functional group. What will be its effect on the overall binding of the ligand with the receptor? Now that is something that we need to estimate. Now intrinsic binding energies. Now we have researchers who have talked about apparent binding energies. Like for example, this researcher called as Page. Okay, he referred to this principle of the difference in the binding of the ligand with or without a particular functional group. Okay, so for instance, if we have a structure, a simple naphthalene structure, let's say I am using the example of naphthalene. Okay, so if we have a simple naphthalene structure that is unsubstituted, okay, and then we substitute, say, suppose position number one with some substitution, position number two with some substitution, position number three with some substitution. So what this researcher did was he went on to calculate the values of certain structures between the unsubstituted structure, that is 1, 2, 3, and all those positions being equal to hydrogen, with the substituted structures. यानी 1 के ऊपर कोई substitution, 2 पे some substitution, 3 पे some substitution, and that is what he went on to empirically determine. Okay, so he called them as apparent binding energies, and apparent binding energies are, you know, related to the substitutions in the functional groups that are done with respect to the unsubstituted. Uh, okay, now this anchor principle, which he said, okay, he called it as the anchor principle. Okay, now the anchor principle assumes that intrinsic binding energies are properties of the functional groups and are independent of the groups to which the functional groups are attached. Okay, so he says that agar chlorine hai, if no matter if I'm substituting chlorine onto naphthalene type of structures or biphenyl type of structures or any other type of structures, we have talked about, for example, benzodiazepines. We have talked about many other structures in medicinal chemistry. So whatever be the basic structure, the Ankel principle states that the intrinsic binding energies are related to the functional group and they are independent of the groups to which the functional groups are attached. Okay, and a core structure can be anything, but functional groups are what are responsible for the binding energies. Okay, and he went on to give out, do many studies and give out data. For example, he estimated intrinsic binding energies for methylene group to be in the range of 12 to 14 kilojoules per mole, for carboxylic acid and amino groups to be in the range of 18 or 28 kilojoules per mole. Okay, he also experimentally gave the contribution of hydrophobic effect, which was calculated to be 0.18 to 0.24 kilojoules per mole. Okay, and solvent effects may complicate the derivation of intrinsic binding energies using intrinsic principle. Now, although the same functional groups might be binding to different structures, may be a part of different structures, but the moment a solvent is introduced, then it changes the intrinsic binding energy. Okay, the moment the solvent is introduced, it will lead to change in the intrinsic binding energies. Okay, and that will complicate the anchor principle, the basic hypothesis which is there behind the anchor principle. Okay, now it has been illustrated by different experiments and those energies can be calculated. Like for example, अगर हमको मिथाइल ग्रुप ऐड करना है, तो मिथाइल ग्रुप चाहे हम लोग किसी भी कोड स्ट्रक्चर, किसी भी बेसिक स्ट्रक्चर के ऊपर ऐड करें, इट विल टेंड टू प्रोड्यूस अ मैक्सिमम ऑफ अराउंड 12 to 14 kilojoules per mole का difference. Okay, 12 to 14 kilojoules per kilojoules per mole ka difference. So that means if we add one methyl group, it is going to change the binding affinity by around 12 to 14 kilojoules per mole. Okay, so that is what is mentioned by Page in his intrinsic, in its anchor principle. Okay, and then active site mutagenesis, we have already talked about this some time back. We have, the protein, the ligand, to the receptor to which the ligand is binding. So if a ligand is binding to the receptor and we change the active site amino acids, then what effect will it have on the binding of the ligand with the receptor? That is something that we need to, you know, check, and that is done using active site mutagenesis. So active site ke amino acids are mutated, they are changed, and then what happens to the binding affinity is checked. So if the ligand binds more better after the mutagenesis is done, then that means the that particular amino acid is not important in binding. Okay, whereas if some amino acid is changed in the active site and the ligand stops binding to the amino acid, that means that amino acid was involved in binding interaction with that ligand. That cannot be changed. Okay, so using active site mutagenesis, we can use this principle of active site mutagenesis to design drugs that can bind better. Okay, so we understand which amino acids are important in binding. So, for example, if it is a charged amino acid, a negatively charged amino acid, which is important in binding, then what we can do is we can design newer drugs which will have a positive charge associated with that part of the ligand which is binding to that part of the receptor. Okay, so that is how active site mutagenesis is important. And average functional group contributions, now this is something again that is related to the average values of the binding energies that are associated with each functional group, similar to something that Page had described in his anchor principle. Okay, so we had Andrew et al. Who in 1984 they calculated the contribution of individual functional groups, and they calculated the binding energies of something like 200 ligand protein interactions in aqueous solution. And they said कि अगर we have a carbon which is sp2 or sp3 hybridized, then it will have a energy difference. It will cause an energy difference of 3 kilojoules per mole in binding. If we have OH group or C double bond O group, then it will cause a binding energy difference of 10 to 14 kilojoules per mole, so on and so forth. Okay, so that is the average functional group contribution principle. Okay, so needs to be stressed that although we have those intrinsic binding energies, different average binding energies that have been calculated, we cannot use it for full-fledged determination of binding energy. So that is what is mentioned over here. All right. Okay. So I think we'll just stop over here, and in the coming lecture, we'll talk about the rest of the concept.